Bienvenidos al canal del grupo de estudio de hormigas neotropicales. El día de hoy tenemos la charla Neotropical Dorymyrmex, Evolution, Biogeography and Taxonomic Implications, a cargo de la doctora Jill Obersky, del Segenberg Research Institute and Natural History Museum in Frankfurt. Síguenos para más contenido relacionado con hormigas y no olvides suscribirte.
Hi, I'm Jill Obersky. I did my PhD on Dorymyrmex, and they're my favorite little group of ants. And I'm going to talk to you today, focusing on the neotropical representatives and their evolution, biogeography, and taxonomic implications. So, given my current audience, I don't really need to try to convince you that ants are important, but we all know that 80% of the biomass of all insects is ants. That's 2.5 million ants per human. So yes, they're everywhere and incredibly diverse, but not all ants are created equal. So species that are tiny or difficult to access and or less distinct can be relatively unknown. This leads to an imbalance in taxonomic attention. Basically, most taxonomic attention is given to visually striking and easily diagnosable groups, but indistinct groups can contain just as much diversity as groups that are flashier, whether that's in genetic distance or life history or all sorts of aspects. These groups can contain cryptic species, for instance, that have been independently evolving for millions of years, all without really changing their external appearance. One example of this phenomenon is, of course, the ant subfamily Dolichoderinae. These so-called odorous ants are chemically really diverse, and they include the invasive Argentine ant that forms supercolonies. They're also very smelly because they're chemically diverse, often compared to blue cheese or banana candy. But in general, Dolichoderines have been difficult to differentiate at the genus level for a long time, never mind the species level. So in general, they need more work than their visibly diverse relatives do. Hot take. One of these underappreciated groups is, of course, Dorymyrmex, which contains 61 valid species, 47 of which are neotropical, and is recognizable by the cute little dorsal cone or spine on their propodium. Not paired, just one right in the middle. Dorymyrmex is widespread in the Americas, and they're very common in dry habitats. Because they favor deserts and grasslands and scrublands and things, they have this distribution that can be called amphitropical, or both north and south of the tropics, concentrated in dry temperate regions. This is in contrast, of course, to the typical latitudinal diversity gradient theory, which focuses on diversity at the tropics. They're also really conspicuous because they have these nests on open ground that look like craters or bullseyes. And so they're a familiar sight to myrmecologists in the Americas. And that makes my life easier because they're frequently collected. But they need some work. So they're fascinating for a variety of reasons. For one, they are agriculturalists themselves. They tend hemipterans and lycaenid caterpillars. Because of this, they're important for human agriculture because they take care of crop pests that we want to eradicate. They contain morphological mysteries like this little cone and spine. The namesake Dory comes from this, but we don't know what it's for. There's behavior and sensory physiology that varies across species. So we've looked into that a little bit, and several species also rely on temporary social parasitism to get their colonies going. So there are lots of different things that make this an interesting group. My personal favorite of the Dorymyrmex anecdotes is that they've been observed throwing little stones down the entrances of neighboring Myrmecocystis nests in order to prevent them from foraging, so the Dorymyrmex can run around and collect all the resources while the Myrmecocystis are being bombarded. I really like this because it's a little bit petty, but they're really neat, but they're still poorly understood. And at the root of all of this is taxonomy, because it's hard to do meaningful studies of broader interest until we have a handle on the taxonomy. And for Dorymyrmex, it's quite complicated. So zooming way out, to To resolve these tricky taxonomic problems and discover diversity, both locally and globally, we need to start at the broadest level, the global scale, and work our way in. In turn, these little ants can teach us about huge processes and consequential things in time and space, like how evolution unfolds and geological changes, like the timing of the Isthmus of Panama. So I'm going to cover about 4 different topics: Dorymyrmex evolution and biogeography, the taxonomic implications of what I've found so far in my research, and tips for identifying Dorymyrmex. So, evolution, here we go. Dorymyrmex diversity is asymmetrical, and what I mean by that is the confounding diversity in the widespread general sense is at the species level. How many species are there? Not really sure. And in South America, there's also been confusion at the genus level, because there are more generalized forms that are in North America and Northern South America, but they've frustrated taxonomists for ages. But in the dry southern parts of South America, there are some groups that look really, really different from each other. They can be spiny and hairy and have long and branching maxillary palps, and these are all modifications for dry sandy habitats. So on the one hand, species. On the other hand, should the southern Dorymyrmex be broken into multiple genera? Because they have in the past. Looking at this historical problem really quickly, since its description in 1866, Dorymyrmex has been divided into as many as 7 genera and subgenera, like here in 1956. But in 1992, Steve Shattuck sank everything back into Dorymyrmex sensu lato. At first, this doesn't seem too complicated if we assume that the genus was divided into 7 and then re-synonymized. But 1956, of course, was just one of many attempts to categorize the genus. So this flowchart from 1866 to 1992 actually looks more like this, where each vertical line represents a published paper changing the taxonomy of Dorymyrmex, and each color representing a different genus. So maybe Dorymyrmex isn't exactly understudied or neglected, because lots of people have tried. The issue is that until recently, taxonomists didn't have the advantages of molecular data. So there's been this whole mess, and now we're back to square one. Everything is just Dorymyrmex. When we find a ground nest, who are we even looking at? What are Dorymyrmex species doing? How are they related? This was the work of my dissertation. So I started with a molecular phylogeny, my happy place. The sampling for this study is shown on the right. I covered the vast majority of the range of Dorymyrmex and did my DNA extractions. 74 of my samples were from the University in Curitiba, University of Paraná, and did my DNA sequencing using targeted genomics, targeting ultraconserved elements or UCEs throughout the genome. Tried to select my taxa and assemble my matrices to maximize geography and diversity in the assemblies. Did partitioning and model selection and completed my analyses under both maximum likelihood and Bayesian frameworks. You can ask me details about those. I'm happy to talk about it. First, a brief primer on UCEs, because they're super common in ant systematics research. Ultraconserved elements, or UCEs, are these really short conserved sequences located throughout the genome, which are great targets for universal primers called probes or baits. And the actual useful DNA sequences we gather are adjacent to those regions, so they're not conserved so tightly. And then I go a step further when I'm evolutionarily modeling, and I model the conserved regions together and the more variable regions together, rather than separating by locus. Separating by evolutionary rate results in a better fit evolutionary model. So on the other side of the analysis, I have a phylogeny, resulting in 69 species-level lineages and a number of undescribed species revealed. So I first started with 32 of the 61 valid species represented that I could identify, and 69 came out the other side. So I have 37 new species that didn't match any existing holotypes or species descriptions that I had access to. Lots of undescribed diversity. But importantly, there are 4 species groups here, each with a different color. So let me introduce you to Dorymyrmex. The 4 species groups that are really robustly supported are here in pink. The flavescens group is Dorymyrmex in the strict sense, Dorymyrmex, OG Dorymyrmex. In green is the tener group, or one time Araucomyrmex, mostly these are from Chile, and the Paramicus group and the Wolfhugeli group together form the former Conomyrma genus. Paramicus group we kind of knew about, but the Wolfhugeli group is new since my last published paper because it's difficult to distinguish from the Paramicus group. Basically, these are all kind of geographically distinct. There's some overlap with Wolfhugeli, and they're kind of morphologically distinct with the spines in the flavescens group and a really big semaphore in the tener group. But the Wolfhugeli and Paramicus groups are basically physically indistinguishable. Despite a significant genetic divergence, there's a reasonably long branch between the blue and yellow clades here. I couldn't find an easy way to separate them. Also, only the Paramicus group has dispersed widely. So it looks like eyeballing the tree and the map here that Dorymyrmex started off in southern South America, made its way northward out of the southern cone, and then radiated out of South America into North America and beyond. North America has its representatives all in one clade, but as we'll see, not every member of this clade is North American. So a tree is great, but what processes are behind this? To start picking away at that question, we need to put some dates on the tree. So I did my divergence dating in the program MCMCtree. And I'll be zooming into this lovely chronogram we see here. On this chronogram, the present is on the right, and crown Dorymyrmex dates to the Oligocene. So the curve on these nodes are confidence intervals showing the age estimates. And crown Dorymyrmex is right here at about 27 million years ago. The age of the crown Paramicus group is about 12 million years in the middle of the Miocene. And because of this general age range for these, it's probable that long-term climate change was helping drive diversification. So some of these climate shifts include the first half of the Miocene being a really cool period where grasslands and seasonal woodlands expanded. And then in blue, here we have the most intense uplift of the Andes Mountains between 12 and 4.5 million years ago, which really reorganized the environmental conditions across the continent of South America, particularly the southern cone region. And then here, the gray bar is the uplift of the Brazilian plateau between 7 and 3.5 million years ago. So this further developed the dry habitats found in Brazil today that Dorymyrmex are so fond of, the Cerrado and so on. So processes like these through the Miocene probably shaped the way Dorymyrmex has glommed on to dry regions, particularly these southern taxa like the tener and flavescens groups, because the crown ages of these clades all post-date the Miocene. So there may have been some intense change happening along those branches. You'll notice this is just the bottom of the phylogeny. So scrolling up the chronogram, we see that Dorymyrmex arrived in North America between 10 and 7 million years ago, right here on this branch. So that was the end of the Miocene. Nifty. I'll get back to biogeography in just a second. Main findings from pulling together a phylogeny. The 3 former genera are phylogenetically supported, Conomyrma with the 2 species groups, Araucomyrmex and Dorymyrmex. But I think evidence is insufficient for splitting the genus because within the context of the subfamily Dolichoderinae, Dorymyrmex is really strongly cohesive. Here you can see its sister to Pharelius. So I don't think any of these warrant new genus status. Great. Number 2, Dorymyrmex biogeography. I wanted to look at it directly instead of just eyeballing the map in the tree. Latitudinal diversity gradients, of course, are one of the best known biogeographic patterns. But there are some groups that defy the tropical most diverse paradigm, instead thriving in temperate environments or even polar climates. So these are the inverse or amphitropical or bimodal diversity gradients and disjuncts. But scientists are finding more and more of these so-called exceptions by the year. Some of examples of things in the Americas that have inverse gradients are Larea creosote bush and lots of other plants, as first described by Raven in 1963. But bees too have an amphitropical distribution. And the recent study of bee biogeography by Orr et al. In 2021 hypothesizes that they're most diverse here because these are xeric areas with high non-forest plant productivity and distinct overlapping faunas, creating a variety of ecological niches where things can slot in and diversify. Another notable group, of course, with this distribution is Dorymyrmex. So this map here is of ant web records, but it's certainly undersampling South America in Argentina, Chile, Peru. The Dorymyrmex is abundant and diverse down here, but ant web doesn't quite reflect that. On the other hand, we know this gap around the Amazon is truly a scarcity rather than a sampling artifact, because there are other genera like Camponotus that have been sampled all over and are listed as such in ant web records. Anyway, amphitropical distribution, I think it counts. I've received some criticism once or twice that it doesn't count as amphitropical because they can be found in Central America, but that's true of bees as well. So when faced with this bimodal distribution with a gaping hole in the middle, the question that comes to my mind is, how are these patterns produced? How and when have organisms dispersed between North and South America? If we're talking about continents, there are 3 main hypotheses, basically. On the one hand, there's the rumored and hotly contested Garlandia Caribbean land span, the Isthmus of Panama, of course, with the Great American Biotic Interchange, and maybe long-distance dispersal, or some combination of the 3. But Dorymyrmex is a great system to test these patterns because they're highly prevalent and frequently collected. And just like ground nesting bees, they have quick dispersal and colonization of new ground areas with their winged reproductives. So main questions here: how did Dorymyrmex develop its amphitropical distribution? Can we reconstruct its path out of South America? And what life history traits have enabled this dispersal? It seems likely that specializing on dry habitats might be contributing to their vegility. So methods-wise, to quantitatively connect these, I started with the Resolve Ecoregions project and discretized North and South America into 8 georegions, basically merging the ecoregions by proximity and similarity of precipitation and vegetation. So taking that plus my list of the current distribution of each taxon and my dated phylogeny, I get hypothesized geographic distributions of the ancestors using a maximum likelihood framework. First question being, of course, did Dorymyrmex reach North America via Garlandia? Short answer is no. So to better visualize this here, this is my overview biogeochronogram. Here we see Garlandia way at the beginning, way before Dorymyrmex is really doing much. So we can rule that out. Then there's a rapid emergence of all of the Paramicus group lineages here and the North American dispersal event shortly thereafter. So here we are zoomed in, Garlandia. This is long before Dorymyrmex began to diversify. So 33 million years ago, Garlandia is out. Of these other ancestors, though, these nodes, you can see the Flavescens group and the Tenaire group ancestors are strongly estimated to have occurred in the southern cone here in pink. Scrolling up this chronogram, we see at the other end, North America was reached before the Isthmus of Panama was closed. Yes, you read that right. So the traditional date of this closure of the isthmus is 3 million years ago. Here, this dotted red line. But the green bar here is when Dorymyrmex arrived in North America, or right here where this asterisk is. So it looks like Dorymyrmex might have traversed the gap before the land bridge closed. So flight over water or a long distance dispersal event, maybe. But I think it's more likely that the growing theory is that there was an early and complex origin to the isthmus rather than a sudden closure at 3 million years ago. It was earlier, more complex. So starting around 10 million years ago, there were long-term shifting islands, and that continued up until the date that we know for sure. So 10 million years ago date is here, this dotted red line on the left. And this one is consistent with the transit of Dorymyrmex. The thing about this asterisk branch, though, is it seems like a big jump. Here we have Dorymyrmex supposedly dispersing from Peru to Baja California, if we go from this to the most closely related taxon. I think this is a little extreme. What's the in-between step here? Because isn't there Caribbean or Central American taxa involved? I did sample those. So let's look at the Caribbean and Central America quick. Caribbean taxa are nested among South American lineages, in fact. So there was some back and forth here around the Caribbean region, but never reaching North America. It was kind of a dead end. But then Central American taxa that I sampled, thinking that maybe it would be in between stages between the continents, actually are nested within the Nearctic clade. So there was some back and forth happening around the Gulf of Mexico as well. There's at least one species that's found in Florida and Texas and Yucatan, Mexico. So we know there was some rafting or something happening around there. So revisiting the Peru to Baja jump, I think this jump is most likely due to incomplete sampling, quite frankly, in Peru and Ecuador, rather than a long-distance dispersal event. But better sampling would be really great, and I wouldn't say no. This is also why we need to do global studies rather than regional studies, because had I only sequenced things in South America, I never would have known that the Central American taxa are actually part of the Nearctic clade. So all of this back and forth is probably because of Dorymyrmex ecology. Basically, Dorymyrmex have grown to specialize on resource-poor environments, things like deserts and scrublands and beaches. I have one collection that is included in my tree, I think, that was collected on an island in the Bahamas at 1 meter elevation above sea level. So they can set up camp basically anywhere, it seems. And because of this, I think it's likely that they used shorelines as dispersal corridors. So even if there were shifting islands back and forth, they could easily hop around to young islands and set up camp somewhere. In other cases, like in Florida, Dorymyrmex is known to have very deep, straight nests, and that might help insulate them against extreme temperatures. We don't know what the nest casts look like for South American taxa, but I would wager that they're definitely using ant architecture to their advantage in the more extreme desert habitats. I think these same life history factors might help explain the timing of Dorymyrmex dispersal relative to other groups too. So Dorymyrmex's northward transit is after that of bees. So here we see Diadasia sunflower bees. They dispersed over water in the earlier Miocene, as found in other studies that I should have cited on this slide. But theoretically, Dorymyrmex has a very similar biology. They nest in open ground, but this pulse of dispersal was too early for Dorymyrmex to be far enough north at the very least. Another comparison point is cephalotes, which colonized Central America several times, but we can't be exactly sure when because of the long branches in the study by Price et al. But I find this interesting because even though Diadasia and Dorymyrmex are ground dwellers, cephalotes, on the other hand, might need shrubs and trees to colonize new areas. So maybe that happened after the open ground nesters. It's fun to speculate about what habitat might have looked like on those shifting Panamanian islands, but it's mostly speculation. There's also one more comparison point with ants, and that's Eciton. So from South America, Eciton colonized Central America quite recently, between 7 and 4 million years ago, which makes sense that it was recent because Eciton has these wingless physogastric queens that are carried by their workers. So basically, they require contiguous land in order to disperse. Both Eciton and Dorymyrmex, therefore, provide evidence that the Isthmus of Panama is actually older than what we've been thinking of it as 3 million years old. So in sum, this inverse distribution was produced by a single dispersal event across the isthmus, but with a little bit of back and forth around the Caribbean and the Gulf of Mexico. And specializing on dry habitats probably has helped Dorymyrmex expand its range. That work is excitingly in review right now. I just got the reviews back, so stay tuned. Next up, taxonomic implications. I made a big phylogeny and Dorymyrmex taxonomy needs work. So, because Dorymyrmex is rapid and ongoing as a radiation, and many of its taxa look alike, we had to rely on this molecular backbone. Thankfully, studying morphology in light of genetic lineages can unlock previously unrecognized diversity. So maybe we can end up with clearly delimited, visually diagnosable species. If we start with the phylo and we reconstruct or recognize species groups, then we look at some morphometrics, maybe we can find some synapomorphies. In other words, if there's subtle morphological variation, it might look like very few species. If things are closely related in a molecular sense, it might look like very few species. But if you combine the two, suddenly all of these patterns start popping out about, oh, these things that look similar are convergently evolved. So taking this back to UCEs, because I've gotten this question a lot, how are ultraconserved elements good for species-level taxonomy? They're really conserved. Well, ultraconserved just refers to the probe, not the resulting sequence data. So the probe adjacent region here in blue is good for deep nodes, and the variable rapidly evolving regions on the sides are good for shallow nodes. So it's good at both deep and shallow taxonomic levels. Basically, if you make your different sequence alignments with different taxon sets, then the best loci are chosen, or partitions are chosen for the set of taxa that you're looking at. If you want a matrix to look at deep nodes, you make your deep matrix. If you want one for shallow nodes, you pick your shallow matrix. If B and C are the shallow ones here, it works out great. So combining all of these things, genetic lineages, morphotaxa, I uncovered lots of convergent evolution, 69 species level lineages, 37 undescribed species, and I even resolved some old problems. Most notable was that the wastebasket taxa, such as Dorymyrmex bruneus and Dorymyrmex geldii, are confirmed to include multiple lineages. So there are 5 or more black arrows here representing things that look like Dorymyrmex bruneus, and there are 3 that look like Dorymyrmex geldii, which surprised me because this is a relatively unique syndrome with the narrow heads and the long antennal scapes and legs. And yeah, geldii might be any of these 3, and one of the bruneus lookalikes is actually in the Wolfhügeli group rather than the Paramicus group. But even though this looks like it's causing confusion, I figured out which of these terminals are the most type-like. So we now have DNA for specimens that closely match the holotypes. On the left, we have bruneus. On the right, we have geldii, which sets the stage for barcoding efforts and will help any future study, basically, that relies on Dorymyrmex species identity. And there they are in the phylogeny. I can point to them. More exciting to me yet was, I think I might have solved the mystery of Dorymyrmex paramicus. So Dorymyrmex paramicus was described by Roger in 1863 with 2 syntypes. In the manuscript, he wrote Brazilian Bahia Corrientes. So presumably Bahia and Corrientes, but Bahia is over here and Corrientes is over here. His 2 specimens are labeled Bahia and Bahia Corrientes. So, given that, I made a few decisions and drew some inferences. I started leaning on this lower specimen, the Bahia Corrientes, and calling that one Corrientes, because it looks more like species from Corrientes that are actually Dorymyrmex thoracicus than it does match the species description in German for Paramicus. So I even had similar individuals represented in the phylogeny. The Paramicus syntype from Bahia is a closer match to the type description. And I had a very similar one here this one was from Mato Grosso do Sul, but there's also some in Pernambuco, which are span Bahia. And then the lower one here, the Paramicus syntype, if I'm calling it not Bahia, only Corrientes, looks exactly like this thoracicus, whose type locality is basically right next to Corrientes. So I'm saying that I've identified these lineages and I'm very excited about it. There are also numerous undescribed species in the phylogeny, such as this beauty from Minas Gerais. And it's listed on AntWeb under the name JTO02, but needs a name because it's particularly distinct and it's super shiny and it's got this interesting elongate mesonotum. And because it comes from Brazil, I joked about this at Bermejo, but I think it holds true. This is the perfect opportunity to finally name a Dorymyrmex species after a Brazilian treasure, the coxinha. It's the perfect name for ants with little cones, and I think that the golden brown color of this particular species makes this ant a great candidate for the name. So the stage is set for taxonomic improvement. We have the DNA. I have some useful morphometrics, lots of holotypes around AntWeb. I didn't open the can of worms of neotropical taxonomy during my PhD, but I'd love to visit South America again, borrow specimens, and formalize some of these IDs. So let me know if you're willing to host me for a visit. Lastly, I have some tips for Dorymyrmex identification. I know you're all super excited to hear them. Basically, it's complicated. So things that make this complicated include interest-specific variation in both color and allometry. Color, you can see here on the right, often intergrades between yellow and brown, sometimes in a way that looks kind of sunburnt, sometimes in a more uniform way. And here on the left, these 2 images are of the same species, and in fact are 2 different paratypes of the species Dorymyrmex smithi. But you can see that one of them has a concave vertex on the head, while the other has a very flat vertex. And this one has a much wider head, and this one has a much narrower head. So a lot of these allometric differences that are used for telling species apart also exist within a species. It's best to use the largest workers of a colony to maximize the differences that you can see between species, which just underlines the importance of collecting entire nest series for social insects, because there is a decent amount of within colony variation. Other tips are to look at the head characters in full face view, but the viewing angle is really important. So by making sure that the frons is closest to you, here between the eyes, rather than the clypeus most close to you, then you get a better view of the vertex and the shape of the head, whether it's wider posteriorly or anteriorly, and so on. I run into this problem a lot with ant web images, is trying to draw conclusions about specimen IDs based on photos is really difficult when some of them are chin up versus chin down. Some useful characters that I found for differentiating taxa in the Nearctic and as well as the Neotropics, although I didn't do as deep a dive into morphometrics there, are head width and length, of course, cephalic index, which is the ratio between those things, interocular distance, the percentage of the head behind the eyes, if that makes sense, as well as the percentage of the head behind the widest point. So how far forward is the widest point of the head? Pronotal CD and mandibular CD. And other characters include the mesonodal profile. In some species, it's very flat. In some species, it's angular. The propodial profile sometimes is concave or sinuate, or of course, as we've seen in the Neotropics, can be a spine or a really elongate cone. And then the cephalic vertex has been really useful. Basically, is it round, flat, furrowed, or concave? So these are things to look out for when you're trying to sort your malaise or pitfall samples to Dorymyrmex morphotaxon. I also have a bunch of photos illustrating species level lineages, which are on AntWeb, but I could compile a PDF guide to the Neotropical morphotaxa like I did for the Nearctic for my PhD. But I have the materials, I just need motivation. So email me if you would find this useful. Real humans are highly motivating. In conclusion, the 4 major species groups of Dorymyrmex were shaped by Miocene climate shifts. Nearctic Dorymyrmex stem from a single recent dispersal event across the isthmus. Integrative taxonomy with molecules and morphology is critical for untangling evolutionary radiations. And UCE phylogenomics can illuminate persistent taxonomic puzzles and find further evidence for major events in Earth's history. I have many people to thank, many funding agencies, UC Davis in particular, lots of places I've visited, many conversations with colleagues and collectors and people who have lent me material. I'm very thankful for all of you. Thank you so much. Thanks for having me, and I would be happy to take any questions.
Thank you so much, Jill, for such an amazing presentation. So now you are totally welcome to send your questions on the chat, or you can also turn on your microphone or camera to ask questions to Jill directly. I will start by asking Jill where people, the contact information is there. So I think they can be free just to send you an email. Yes?
Yeah. Please get in contact. I'll just put that in the chat.
Yeah, that will be exciting. So we have some comments. So Roberto Jose Guerrero says, thank you very much, Jill, for such a wonderful presentation.
Thanks, Roberto.
It's so amazing because finally we have some tools to identify that, and it's important. So one question that Roberto mentions is, Argentina is the land of the Dorymyrmex. Surely with the Kuznetsov collection as the first repository for these ants. What were those problems in accessing the Argentine Dorymyrmex?
Yeah, so I had originally gotten in contact via email with Fabiana Coeso, and she was open to the idea of collaboration, willing to host me, which was great. I applied for a small grant to visit. I was accepted, which was great. And then I didn't hear back from her again, and I didn't know other Argentinian myrmecologists to reach out to. So because of scheduling issues, I connected with Rodrigo Feitosa in Curitiba, Brazil, and made a visit to the museum in Sao Paulo, as well as the university in Curitiba, and based my loans on more Brazilian taxa than Argentinian taxa.
Yeah, thank you. Thank you, Jill. I think if there are like any people from Argentina here that can be contacted, it will be wonderful. We can also make an open call if you want, asking for Argentina colleagues to stay in touch with you.
Yeah, I would love to visit and do some work with the more wild looking Dorymyrmex.
That's amazing. So we have more comments on the chat, and one of them is for Giampiero. So he says, thank you very much for this talk. For the biogeography, did you use BioGeoBEARS or are there other alternatives?
Yes and yes. I did use BioGeoBEARS, and there are other alternatives, including GOC, which is in the same class of models as BISI and Classy, and if you've heard of any of these. But I am not as well versed in them, so I can't say much about them.
And Natalia Ladino says, this presentation was fantastic. Thank you very much. Out in the field, which is the best technique to collect these ants? Males and queens are easy to associate to workers.
Good questions. So in the field, I think the best way to collect Dorymyrmex is probably spotting the nests and getting nest collections right there at the entrance. Some nests have multiple entrances, especially in the Nearctic region. There are polydomous species, but getting nest collections right at the spot so you can be sure that things are from the same colony, and you can account for within colony variation and measurement is super helpful. Males and queens are sometimes easy to associate to workers if there's a really unique morphology in the workers. For example, Dorymyrmex elegans is super elongate, has like an egg-shaped head, very long scapes, very long legs, and those characteristics kind of hold true for the males and queens as well. But ideally, of course, you collect them together, which is difficult to do. So I've found actually a helpful way to do that is to talk to your local ant hobbyists, because there are people who are ant keepers that keep ants as pets, and they have a handle on which species are having mating flights when, because that's when they go and start their colonies. So knowing when the mating flights are happening, maybe priming the nest with some water the previous night, and then digging at the surface the next day is the best way to gather your full set of casts.
Yeah, that's very important, especially when you only have males or queens in collections, but no workers.
Yeah. Otherwise, sequencing, of course.
Yes. So Natalia says, haha, cool. Already motivated to collect. Great, Natalia, thank you. And Roberto Jose says, is like another question. The results of Dorymyrmex colodi is incredible. 3 independent lineages. The morphology of that species seems unique without the possibility of error.
I collected some Dorymyrmex in the savannas of northwestern Colombia. I sent you a couple of years ago. Like Dorymyrmex goldii in the configuration of the propydium. But when analyzing the length of the escape and thickness of the mesosome, I was able to discern that it did not correspond to goldii. So do you think this could be the result of previous misidentification of those specimens?
That's a good question. The short answer is yes. When trying to discern species based on the propydium within the Paramicus and Wilfuguli groups, I had lots of trouble because there's so much variation in the propydial profile. But based on head shape and scape length and eye size also is very important. I do recall looking at your specimens and sequencing a number of your specimens, actually, and they were not coming out with the Geldii that's the most type-like, but were rather coming out as something new from Colombia. There were some that you had called your species number 2 that I think actually were closely related to Biconus. There are some intricate details here that I'd have to go check my notes for, but I'd be more than happy to email with you about this because I did sequence a handful of your material and they have made it into this final big phylogeny.
Yeah, that's great. So I guess you can contact Roberto directly. And he also has a follow-up question. He says, the last question of many, and yeah, he has many questions, so feel free to reach out to you, is any idea about Colombia in the biogeographic pattern of the genera in Northwestern? We have several Dorymyrmex in our collection in Santa Marta. You are welcome if you would like to visit us.
Yes, I would love to visit Colombia right there at the base of the isthmus, get some clues on what's happening there. All of the taxa I sequenced from Colombia are nesting within the general South American lineages of the Paramicus group. So they weren't necessarily any closer to the Peru hook to the Nearctic. But I can send you phylogenies and things later. And yeah, I'd love to visit Colombia. I'll let you know if the doors open for that possibility.
Great, that will be amazing if we can arrange an encuentro myrmecológico in person. So we can have you there. Great. Victor Hugo Guzman has a question. So they ask, why barriers for distribution had to ecology or phylex in tropics for Dorymyrmex? I can send you a question directly. Okay, this is a question.
That helps. Thank you. I was searching for it. Barriers for distribution, ecology. Well, yeah, it's interesting because the Dorymyrmex that have radiated, that is the Paramicus group, are more generalist than the more southern taxa. So they are more tolerant of more humid environments. But I was looking at the Amazon basin as a barrier, basically, and thinking about this in terms of the ants would either have to go around or maybe island hop in dry patches. And then I started looking at maps of the vegetation changes during the last glacial maximum and that sort of thing. And there's so much change in the extent of the rainforest versus the grasslands, even within the past 22,000 years, that looking at ages like 3 million years for Dorymyrmex there's so many changes that could have impacted the dispersal of Dorymyrmex just because of like smaller scale climate and vegetation shifts that, yeah, sometimes I question whether the big geological changes like continents shifting and isthmuses closing are really the ultimate explanation for things, when I think it may be shorter term processes that are really happening, quick change and quick dispersal and retreat by Dorymyrmex. I don't know.
Thank you. I think you mentioned it in the talk that the ecology, there will be a recent paper published by you about the ecology of Dorymyrmex, so that we need to stay tuned. Yes, just stay tuned for new papers. That will be excited to know more about the ecology. We have a last question, and it is from Diana Corky, who says, Jill, thank you very much for your talk. Recently, I have become acquainted with the concept of extended synthesis, and I'm now trying to view evolutionary processes from a perspective where individuals be more involved in their own evolutionary process. Do you think any of this could be possible within the work you have done?
I think so. I'm not that familiar with extended synthesis, but there does seem to be a decent amount of queen choice when it comes to colony founding, both in terms of dispersal and also to social parasitism things, which are better known from the Nearctic. But individuals within a colony, meaning the workers, or individuals like colony founders, but I'm not sure how to parse the question, and I don't know enough about extended synthesis, quite frankly.
Great. And we have a comment from Victor Hugo Guzman, who says, thank you. All right, vegetation is progress for entomological prospects in insects grassland communities. Great. I have a last question, but if people want to send other questions, I will read them. But my question is more related to the resolution that we can get with the molecular data that you're getting. So in South America, accessing to sequencing for like ultraconserved elements is quite hard, but maybe accessing the metabarcoding is easier. Do you think you can find resolution at the metabarcoding level to distinguish a species within the genus Dorymyrmex?
Metabarcoding, like taking a sample from a pitfall trap, or?
It's more like if you use one molecular marker, like CO1, instead of use small parts of the genome, like ultraconserved elements, but you can use only like CO1.
Right. I guess I'm wondering, do you mean barcoding in general, or do you mean metabarcoding?
Both of them.
Okay. Yes, I think it's definitely doable. Something that I've been meaning to do actually is reach out to you about Galapagos Dorymyrmex and putting a whole bunch of CO1 sequences together, because that's easy to pull from UCE data. So then we can have the same data format and just integrate everything, and it'll be great. I do sometimes see different results doing a full UCE set and that same set of taxa with just the barcodes, different topologies of the trees. But in general, I would say, yes, barcoding is feasible.
That's great. I think that's a relief for people who, we can only access, most universities can only access to barcoding instead of the whole genome sequencing or UC. That sounds great. So thank you so much for being here, Jill. I think there are plenty of opportunities for people to contact you to talk about projects. The contact information is here, but also Jill will share the contact information here on the chat. And I think if people reach out to us asking for your contact information, we'll share it as well.
Yes, please.
Thank you so much, and see you in the next Encuentro Myrmecológico in July.
Okay. Thank you, everybody.
Bye.